Hey, so what I'd like to do today is talk to you about transcription and translation. Transcription and translation is the process of expressing genes so that they can be turned into RNA, and that RNA message can be used to create a protein. So we'll start with a gene. This gene here is just a model of what a gene would be. It's made up of the A, T, C's, and G's, nucleotides, and this gene is double-stranded DNA. So you'll see there's a 5' end on one end of the gene and a 3' end on the opposite end of the gene, and that top strand has a complementary strand that's beneath it, and that complementary strand is anti-parallel to the top strand. So that complementary strand starts with a 3' end on the left side and ends with a 5' end. One thing we know about that DNA is that those two strands of DNA are complementary, and they're complementary to each other, and they're connected by hydrogen bonds. That means that the A, T, C's, and G's that you see on the screen are actually connected, meaning we can disconnect them. And so when transcription is going to happen, that means we need to take the DNA that we have and break the hydrogen bonds and start reading that DNA and turning it into RNA. The machinery that will do this need to rely on a couple of features that are in the DNA to know where to begin transcribing a gene. The two features that the machinery will look for are this TA region, which is called a Tata box, and transcription factors, which are proteins, will use this sequence-specific region of DNA to bind to it. And there are also other proteins called enhancers that will bind to another region of sequence-specific DNA that is upstream of this Tata box. And it's through the binding of the proteins that are called enhancers to the enhancer region and transcription factors to the Tata box that the enzyme that will be transcribing the gene downstream, that means these letters to the right of the Tata box, the enzyme that will be doing that is RNA polymerase. And RNA polymerase will only begin transcribing this gene that is downstream of the Tata box if the enhancer is bound and the transcription factor at the Tata promoter site is also bound. So once the Tata box is bound with its transcription factor protein and we have the enhancer bound to the enhancer site, RNA polymerase can begin by breaking the hydrogen bonds of the double-stranded DNA. And as it breaks the hydrogen bonds of double-stranded DNA, it is able to read one of the strands of that DNA. And so what you'll see here is I took the double-stranded DNA that was connected by hydrogen bonds and I separated it. What you'll see here are two labels for this top strand and for the bottom strand. The top strand we call the sense strand, and the bottom strand we call the antisense strand. Some other words we use to describe the top strand is the coding strand, and some other words for the antisense strand, which is the bottom one, are the template strand or the non-coding strand. And these terms can be a little bit tricky, but here's what I'd like for you to know. When RNA polymerase begins transcribing the DNA message, it needs to read the template DNA. And remember, template DNA is this bottom strand. It needs to begin reading the template DNA, moving on the template DNA from the 3' end of the template to the 5' end of the template. And so it is moving and reading this bottom strand from left to right. And as it is reading the template strand or the antisense strand or the non-coding strand, it is pulling RNA nucleotides and it is taking those RNA nucleotides and it is matching up the DNA antisense strand or the template strand with a complementary RNA nucleotide. So as you'll see, a C is on the DNA and a G is being pulled in for its complementary. You'll also notice that when there's an A in the antisense strand,
it's 11:30.
We add a U to the mRNA strand, and so forth and so on until we get to the end of the message. And once that mRNA message is produced, we will have that mRNA message meet a ribosome. And there are two parts to this ribosome. There is a large subunit to the ribosome and a small subunit to the ribosome. And this mRNA message will run through the middle of this ribosome. And as it's being pulled through this ribosome, tRNA molecules, which are RNA molecules that have an amino acid attached to one end of them, will meet up with this mRNA message as it's being pulled through the ribosome. And if this mRNA message and the tRNA message bond, then an amino acid can be added to a growing chain of a polypeptide that we're producing. But in order for that to happen, the ribosome can only begin translating this RNA message by starting at a thing called a start codon. And a start codon will always be the letters A, U, G. And so what we have to do with this mRNA message that we produced is determine what's called the reading frame of the mRNA. And the reading frame is the frame that the ribosome uses to produce the amino acid sequence. And so if we were to take possibility 1 and we were to separate possibility 1 into groups of 3 letters of nucleotides called a codon, we will then check and see if that message contains an AUG. Because if the message does not contain an AUG, then we know that the ribosome can't use that reading frame as a way of translating the mRNA message into an amino acid sequence. So let's try it. I'll take 3 letters at a time, and I'll separate them. And let's check. Do we have AUG? No. So this message cannot be used. Let's make it light gray. Let's check. Whoops. Let's check this message. So we'll do 2 letters at a time. Let's start with 1 letter at a time, sorry. So 1, and then 1, 2, 3, space, 1, 2, 3, space, 1, 2, 3. Let's see if there are any AUGs. And possibility 2 does not work. There are no AUGs in this message. And lastly, we'll do 2 letters at a time. 2 letters at the beginning. And let's space this out. And we see great. Possibility 3 begins the message at the 5' end with an AUG. And so we know that possibility 3 is what we can use to translate this message into amino acids. So here's what we have to do. I'm going to erase this stuff here. Okay, so we have our mRNA message. We know the reading frame, so we have the reading frame split out. This message is being run through the ribosome, and it's being pulled through the ribosome so that the 5' end of the mRNA message goes through first. That means that this mRNA message needs to link up with the tRNA message that's complementary to the AUG you see at the beginning. And so the only letters that are complementary to that message are UAC. And this, sorry, I'm gonna move that. This tRNA that you see in the screen right now is carrying an amino acid. And I'll tell you how to determine what amino acid that tRNA is carrying in 1 second. So what we'll do is we'll have the mRNA message being pulled through the ribosome. And as the mRNA message goes through the ribosome, these tRNAs will also come into the ribosome. And as a little bit more of the message goes through the ribosome, another tRNA comes into the ribosome and hydrogen bonds with the mRNA message. And so what we'll see here is the assembly of all of these amino acids in a long chain, and until we get to what's called a stop codon. And a stop codon comes in 3 flavors. The letters of mRNA can be UGA, they can be UAA, or they can be UAG. And anytime you get to those 3 letters, you can have UAA, UAG, or UGA. And in this case, we have UGA. That signifies that we have what's called a stop codon, and the message has to be, the translation has to be stopped. We can't continue moving forward. The ribosome falls apart, and our polypeptide or amino acid sequence is finished. So let's figure out what those amino acids actually are. So in order to do that, what I'm going to do is just label the amino acids that we have. I'm going to use just blue. And what we can do is determine what the amino acid sequence will be. Here's the first thing. We need to know that as the amino acid is being made, the N terminus will be the end that leaves first. And the C terminus will be the end of the amino acid sequence that leaves last. When the ribosome is reading the AUG, the only tRNA that it can bind to is UAC. And we're going to use this table, this lookup table, to see exactly what amino acid the tRNA is carrying. And if I have an mRNA sequence with AUG, that means the tRNA is carrying a methionine amino acid, and that is the start codon. So the first amino acid that we'll have in the chain is methionine. And then what we can do is go to the next codon, CUG. CUG, so we'll go to the first letter C, UG-leucine. So this is the second amino acid. The third amino acid is CGA. CGA is arginine. It's the third amino acid that we're adding. The fourth codon is AAC. So we're going to find AAC, asparagine. We're going to add that. And the fifth one is UUU, and that's phenylalanine. The sixth one we have here is GGC, and so we'll find that GGC is right here, glycine. Remember, when we get to UGA, UGA is a stop codon, so the ribosome cannot continue synthesizing, it falls apart, and no amino acid is added at that point. So we're finished the amino acid sequence. We have a methionine, peptide bonded to a leucine, peptide bonded to an arginine, to asparagine, to phenylalanine, to glycine, and we're done.